You know, every single living thing, from the tiniest bacterium to you and me, it all runs on an instruction manual, DNA. But how in the world does life make a perfect copy of that manual, billions of times over, every single day? Today, we're diving into the incredible molecular machinery behind life's ultimate copy job.
Okay, I want you to imagine something. Imagine trying to copy a massive library of books, letter for letter, in just a few hours, and doing it billions of times without a single mistake. Sounds impossible, right? Well, that's exactly the challenge that every living organism on Earth faces. How is this staggering feat even possible?
Well, the answer is it's so elegant. It's a process called semi-conservative replication. See, instead of trying to make a brand new copy from scratch, the cell does something genius. It unwinds its DNA and uses each of the original strands as a perfect template. And what do you get? 2 new DNA molecules, each one a perfect hybrid of old and new. It's nature's way of making sure nothing gets lost in translation.
All right, so here's our game plan for this explainer. First, we'll meet the tiny molecular machines that make all this happen. Then, we'll compare the high-speed approach of bacteria with the careful marathon happening inside our own cells. And finally, we'll get into why these tiny differences are a huge deal for fighting disease.
Okay, let's dive in. DNA replication isn't just one thing happening. It's more like a massive construction project going on inside our cells. So, let's meet the crew, the highly specialized team of molecular machines that gets this incredible job done. You gotta understand, every single member of this molecular crew has a super specific, critical job. First up, you've got helicase. Think of it as the demolition expert. It literally unzips the DNA. As it opens up, these things called SSB proteins jump in. They're like support beams, holding the strands apart so they don't just snap back together. Okay, next, primase comes along and lays down a little RNA primer. This is basically the starting block, like a chalk line for the builders. And then the star of the show arrives, DNA polymerase. This is the master builder, synthesizing the new strand, letter by letter. And to finish it all off, you have DNA ligase. It's the welder, sealing up all the little gaps to make sure the new copy is absolutely seamless.
So that's the team. But here's the thing. Not all life uses this toolkit in exactly the same way. Let's start with the prokaryotes. We're talking simple organisms like bacteria. For them, it is all about one thing: speed.
See, their whole genetic blueprint is just one single circular loop of DNA. Super simple. The copy process kicks off at just one starting point, called ORI-C, and then it just races in both directions around the circle until the 2 construction crews meet on the other side. It's unbelievably efficient.
And when I say fast, I mean it's mind-blowingly fast. We're talking about 1,000 nucleotides, those are the letters of the code, every single second. That kind of speed is exactly why bacteria can divide and multiply like crazy.
All right, so that was the sprint. Now let's switch gears and talk about a much more complex system, the one inside you and me. For eukaryotes, that's us, replication isn't a sprint. Oh no, it's a very carefully controlled marathon.
So instead of one simple circle, our DNA is packed into multiple long, linear things, our chromosomes. Now, to copy all of that information in a reasonable amount of time, our cells can't just start in one place. They have to start in thousands of places all at once. It's like firing up thousands of copy machines at the exact same time. But of course, there's a trade-off. Our polymerases, they're way more methodical. They only add about 50 nucleotides per second. Why so slow? Well, that slower pace is part of a whole system of checks and balances. You absolutely need that when you're trying to accurately copy a genome as massive and complex as ours.
And this brings us to a really unique problem that only eukaryotes have. Because our chromosomes are straight lines, not circles, the copy machine can't quite get to the very ends, the tips. These tips are called telomeres. So with every copy, they get just a little bit shorter. To fix this, we have this amazing enzyme called telomerase that comes in and rebuilds the ends. It's essential for stopping us from losing critical genetic info.
Okay, so we've got the bacterial sprint and the eukaryotic marathon. Let's put them side by side now and really break down just how different these 2 strategies are.
When you boil it all down, it really comes back to organization. A prokaryote has this one tidy circular blueprint. A eukaryote, on the other hand, has this enormous library spread out across multiple linear books. And that one fundamental difference in structure, it dictates absolutely everything else about how they copy their DNA.
So this table really sums it up perfectly. You can see the trade-offs right here.
Prokaryotes, they are built for speed. They've got a single origin on a simple circular genome, which lets them replicate incredibly fast. But then you look at us, the eukaryotes, with our many linear chromosomes. It's a totally different ballgame. We need tons of origins just to get the job done in time, and we need that special enzyme telomerase to protect the ends of our DNA. All this complexity means we have to go slower, be more deliberate, and even use a whole different set of polymerase enzymes.
Now, I know this might all seem like some tiny molecular detail, right? But trust me, understanding these differences is the key to some of our most powerful medicines. It also gives us some incredible insights into really devastating diseases. Because we know these differences, we can design drugs that are surgically precise. Take antibiotics like quinolones. They go after an enzyme called DNA gyrase, which bacteria absolutely need for replication, but we don't. So it stops the infection cold without hurting our cells. It's the same idea behind some chemotherapy drugs and antivirals. They work by jamming up the replication machinery of just the cancer cells or the viruses, while leaving our healthy cells mostly alone. But what happens when our own super complex machinery fails? Well, the consequences can be pretty severe. For example, if there's a defect in that helicase enzyme, the unzipper, it can cause something called Bloom syndrome, which leads to all sorts of replication mistakes and a really high risk of cancer. And those telomeres, the ends of our chromosomes that get shorter, that process is directly tied to aging. And here's a spooky twist. Many cancers actually figure out how to switch that telomerase enzyme back on, giving them a kind of biological immortality that lets them divide forever.
So just let that sink in for a second. Our very own DNA has a built-in counter, a kind of aging clock. And this clock ticks down a little bit with every single cell division. It's this constant reminder of the incredible balancing act that's required to maintain our complex code.
And that, of course, brings us to a huge, profound question. This one enzyme, telomerase, seems to hold the key to both aging and the immortality of cancer. So what if? What if we could learn to control it, to safely turn it down in cancer cells, and maybe, just maybe, turn it up a little in our healthy ones? Could we actually hold the key to a longer, healthier life? It's an incredible frontier of medicine, and it all starts right here, with understanding this simple tale of two copies.